Now the call is being recorded, and now it's over to you, Organizing Secretary Dr. Atul Preti, sir, please.
Thank you, sir. On behalf of GPGC Bajpur, SS College Shahjahanpur, entire organizing committee, and on my own personal behalf, I welcome Dr. Ahmad Ali from University of Mumbai. Thank you very much, sir, for sparing your valuable time with us on such a short notice. And now I call upon Dr. Nirupama Dalakoti to formally introduce our guest to our audience. Over to you, Dr. Dalakoti. Thank you.
Thank you. Good morning, everyone. I welcome you all in the fourth day of invited lecture series on Biological Science 2020, organized by GPGC Bajpur, SS College Shahjahanpur. I am honored to introduce our eminent speaker for today's session, Dr. Ahmad Ali, Assistant Professor, Department of Life Science, University of Mumbai. Dr. Ahmad Ali has 13 years of teaching and research experience, and Dr. Ali has vast experience in publication and published 35 papers in journals of international and national repute, and also published one book. Dr. Ahmad Ali has won many awards like MBO and CSIR Travel Grant, Best Oral Presentation Award, and several fellowships. Dr. Ahmad Ali is also involved in 5 projects of UGC. Sir, we are very privileged to have you on this series. Now I request Dr. Ahmad Ali to deliver his lecture. Sir, please.
Yes, thank you, Dr. Nirupama, for your kind introduction.
Welcome, sir.
Yes. At the outset, let me thank Dr. Adesh Pandey sir and Dr. Atul Preti sir for inviting me to deliver a lecture in this ongoing series, which is third among that the invited lecture series. So they had very eminent speakers and very diverse topics. To I must congratulate all the colleges and this Government PG College, Bajpur, SS College Shahjahanpur, and Society for Research in Biological Sciences for coming together and having such a wonderful series of lectures. This is third, and in the third, mine is the fourth lecture. So it's really wonderful to see the participants and eminent speakers. As I could see the list, there have been lectures on biotechnology and other things. My lecture will be slightly more technical than others. So I hope I'll be able to make it understand to the UG and PG students, because it's meant for them as well. So if there is time, I would like to talk about my research also, but let me talk about what happens during, what will be the biochemical events during the process of this. So again, with thanks to Dr. Adesh Pandey and Dr. Atul Preti and all the organizing committee members, I'm going to start my lecture.
Yes, sir. Sir, please go ahead.
Yes, sir. Are you able to see my slides, sir?
No, sir. It is not sharing even.
Sir, is it visible now?
Yeah, it is visible.
Yes, sir. Now, yeah.
Okay. Okay. Thank you, sir.
You can enlarge this slide even.
Yeah, yes, sir. I am going to do that. I just wanted to check that. So, is it now?
Yeah, it is okay.
Yes, sir. So the topic is sugar-induced changes in biomolecular structure during diabetes. So the diabetes term is not new to all of us. Sugar, when we use the term in biochemistry sugar, we mean glucose and those sugars which are sweet in taste, like monosaccharides and disaccharides. So this sugar molecules are very beneficial to our health, or important. We cannot survive without them. But as we know that if there is excess of this or extra amount of this sugar present in our body, it can cause more harm than the benefit. So this is one example. I'll be discussing about this mainly. So these are the complications which arise due to excess consumption or underutilization of the sugar which is there in our body. So I'll be talking about, this is just a cartoon to show that what happens and what are the symptoms, what are the complications, and how do we prevent this. So this will be the overview of my lecture, where I'll be briefly talking about biomolecular interactions, then about diabetes, and then glycation, and finally I'll focus on advanced glycation end products. So I hope if at any point, if it is not clear to students or research scholars, please do make a note of this, and you can ask this during the question answer session. So we all know that metabolism is basically an interaction between the molecules or biomolecules, and metabolism is very important, or biomolecular interaction is very important for driving the life processes. It makes possible for the generation of energy and produce raw material which are required for synthesis of larger molecules and carrying out the other work of the body. So when all these biomolecules are proper, they come together and lead to a proper cellular function. However, there are chances, there are incidences of errors happening in this biomolecular reactions, and these interactions, these reactions, or these errors lead to fatal consequences for the organisms. These consequences may be due to the toxic accumulation of reaction products in the cell, damage due to oxidation, deficiency of an enzyme or protein, and overstimulation of certain reaction pathways. So there are many of this kind of reactions, like you have what you call oxidation, you have nitration, and there are many other. I'll be focusing now around one event which is related to sugar, and this event is known as glycation. So very simple terms, glycation can be defined as a non-enzymatic aminocarbonyl reaction between sugars and proteins, lipids, or nucleic acids. So we all know that there are several sugar residues which are very important for our metabolic processes, like fructose, glucose, ribose, and xylose, and so on. But the most important sugar which is there in very high concentration in our body is glucose. So whenever we eat, we have in our food sources, we have carbohydrates, proteins, and lipids. So we all know that the main source of energy for our many of the work and instant energy source is carbohydrates. And carbohydrates, when we say carbohydrates, most of the carbohydrate or polysaccharides have glucose as the major constituent. So glucose is the most abundant monosaccharide available as food, and thus it becomes most important molecule for most of the living organisms. So this is the structure of this glucose molecule. I have shown it because when we discuss the glycation, it will be important to realize the. So for a healthy condition of the body, the balanced metabolism of glucose is very important. But there are incidences, there are events where minor fluctuation in the metabolism happens, and the function of many organs get jeopardized and lead to a disease condition. As far as glucose metabolism and disease or fluctuation in the metabolism is concerned, The two consequences which are seen very commonly are hypoglycemia and hyperglycemia. Hypo means less concentration, less amount of glucose in the blood, and hyperglycemia means high concentration of the blood in the glucose in the blood. So this is a cartoon which shows that what happens during hyperglycemia and what happens during the hypoglycemia. I'll not be focusing on this because I would like to spend more time what happens when there is a high level of sugar in our body. So we all know, and diabetes has become a very common health issue in our all the populations of the world. So every year we are seeing an increase in the number of people affected by this disease that is costing our health system.
Excuse me, sir. I think you have muted yourself somehow. We are not able to listen. Slides are clear, however.
Okay, now I'm clear.
Yes, sir. Yes, sir. No,
And
sir,
my
yes.
slide is visible.
Yes, slides are visible. Yes.
Okay, sorry for. Please do let me know if something like that happens, sir. Thank you, sir.
Yes, sir.
So, yeah, pardon, sir.
We are very much here, sir. Any problem, we'll let you know. No
Yeah, thank you.
Please tell me.
So, yeah. So, half of this population of diabetics is living in India, China, US, Brazil, and Indonesia. Some of these countries are still developing countries. And a rough estimate says that out of 100 diabetic people, 30 people live in India at the global scale. So this is a report from our review that this shows that how the trend of diabetic is increasing, and you can see that in 1995 we had 100 million, and by 2045 it may cross 600 million, 600 million of the population. So as far as the relationship between diabetes and glucose is concerned, it's in very simple terms, biochemists or doctors define it as either it originates from or manifests the dysregulation of glucose metabolism. And one of the most it is one of the most prevalent metabolic disorder, which also comes under the classification of non-communicable disease. So it occurs as a result of the inability of pancreas to produce insulin, that's known as type 1 diabetes mellitus, or cells to effectively utilize the produced insulin, that is known as type 2 diabetes mellitus, which is due to the inefficiency or insulin insufficiency or insulin resistance. So if you see the whole process of diabetes and the consequences, it is manifested as a result of inefficient metabolism of carbohydrates, fats, and proteins. So we can say that it's not just because of the carbohydrate metabolism, it also includes the impairment of fats and proteins. So we can see the consequences or manifestation of this as obesity, nephropathy, hypertension, dyslipidemia, non-alcoholic fatty livers, and systematic inflammation. So if you can summarize it, the diabetes and its secondary complications, they are basically because of some of the cellular events like oxidative stress, reductive stress, altered lipoprotein metabolism, carbonyl stress, increased protein kinase C activities, ages hypothesis, and high bone mineral density. So these are all cellular events which lead to the consequences of diabetes and its secondary complications. So if you would like to summarize that the type 2 diabetes mellitus can happen because of the inflammation, oxidation, glycation, or toxicity, or the type 2 diabetes mellitus can lead to this. So they are interrelated to each other. So how do we manage diabetes mellitus? We inhibit the carbohydrate utilizing enzymes like amylase and glucosidase, activation of AMP kinase, inhibition of glycosylation of hemoglobin, modulation of glucose uptake, inhibition of aldose pathways, advanced glycation in products formation inhibition, and enhancers of insulin secretion and pancreatic beta cell proliferation. So depending on the condition of the pathogenicity of this diabetes mellitus, the approaches can be used for this. So as we mentioned that the major consequences of impairment of glucose are hyperglycemia. So major symptoms of diabetes include hyperglycemia and hyperlipidemia. So what happens when there is hyperglycemia and hyperlipidemia? These induce inflammatory response like glucose autooxidation, induction of polyol pathway, synthesis of prostanoid and glycation of proteins, which further lead to oxidative stress by the generation of reactive oxygen species or free radicals, for example, superoxides, hydroxyl radicals, and so on, and negatively affect the antioxidant defense system, what we call as enzymatic and non-enzymatic defense systems, which include superoxide dismutase, catalase, glutathione, and so on. So if the hyperglycemia is there in the body for a long time, it leads to deterioration of blood vessels and causes damage to vital body organs like kidney, nervous system, eyes, and heart. What in the medical terms people call it as retinopathy, nephropathy, and cardiopathy. Okay. So the intensity of this hyperglycemia can be manifested in two forms: macrovascular, like heart attack, stroke, and peripheral vascular disease, or microvascular, for example, retinopathy, nephropathy, neuropathy, and so on. So, because of this hyperglycemia, another condition is seen that is known as glucotoxicity. Glucotoxicity means an abnormal level of glucose which becomes toxic to our body. So this abnormal accumulation of glucose in the body leads to accumulation of undesired products in the body. So because of all these studies and researches on diabetes, the modern theory of diabetes focuses on oxidative stress and glycation and how these can be prevented. So you can see that under diabetic hyperglycemia, what happens at molecular level, the proteins or vital proteins in the body, they get modified by one of these mechanisms. For example, protein carbonylation, where a carbonyl group is added to the protein nitration, a nitrate group is added to the protein succination, protein glycosylation, protein smolylation, protein acetylation. In conclusion, any of these modification to the vital proteins in the body in diabetic hyperglycemia lead to the improper functioning of the body proteins. And then we realize, or then the person realizes the consequences of diabetes, what we call as secondary complications of diabetes. So I'll be focusing on glycation because the theme of and it's not possible to cover all these modifications in one lecture. All of them have their own mechanisms, their own consequences, and so on. So as far as sugar's direct interaction with a protein is concerned, it is glycation. So it's the most significant process of protein modification mechanism in the diabetes, which is manifested because of the reductive nature of glucose. So we all know that, and we also use this chemical property of glucose to estimate glucose in the body, that glucose is a reducing sugar. So glycation can be defined as a spontaneous non-enzymatic amino-carbonyl reaction between sugars and long-lived proteins, lipids, and nucleic acids. So basically what happens is that there is an interaction between the carbonyl group of the sugar. Where is this carbonyl group present in the sugar? It's the C1 of the glucose and C2 of the fructose. So aldose sugars have it at C1 position, and keto sugars have it at their C2 position. So this carbonyl group in the form of aldehyde or ketone is very reactive. So they react with the amino group of any other nitrogenous compound like proteins, lipids, or nucleic acids, or any other protein. And this process is non-enzymatic. Why the term has been non-enzymatic stressed here? To differentiate it from another process which happens in the endoplasmic reticulum and Golgi bodies, what we call as glycosylation of the protein, which is part of a post-translational modification of the proteins. So when the proteins are synthesized in the endoplasmic reticulum, they undergo this post-translational modification where sugars are attached, and that process is known as glycosylation. And that is under the influence of enzymes. This process is non-enzymatic. It does not involve any enzyme in the process. And the process is irreversible. So we can see in the blood capillary, we have different proteins like albumin, we have hemoglobin. So these proteins, they interact with a D-glucose sugar, and the interaction is very specific. The carbonyl group of the sugar and the amino group of this protein, they interact, and the interaction is initially very fast. It leads to a formation of CIF base, that is a reaction between the amino group and the carbonyl group or aldehyde group. This CIF base is formed, which again undergoes modification. That's a slow process that leads to a ketamine that is quite stable, and further modification of these ketoamines, which are also called as early glycation products. I will discuss later also. So this ketamine attachment to the hemoglobin or serum albumin leads to the improper function of these proteins or any other protein which get glycated, what we call as formation of glycated proteins, which simply means attachment of a sugar to the protein non-enzymatically. So we can see here that glucose and fructose, the two very important monosaccharides, they have this glycolytic pathway. They get converted to dihydroxyacetone phosphate and glyceraldehyde 3-phosphate, which get converted to, under excess conditions, they get converted to dicarbonyl precursors, which are commonly known as glyoxyl or methyl glyoxyl. And further modification of this glyoxyl or methyl glyoxyl leads to the formation of advanced glycation end products, that is the terminal stage, which I'll again, and this again leads to or binds to RAGE. And so these can be several mechanisms by which metabolic diseases are seen in the body: lipogenesis, sphingolipid metabolism, and so on. So again, a bigger picture of that: glucose oxidation, autooxidation, or lipid peroxidation. These are the very common reactions which happen in the body, or in the normal healthy people also it happens. But the body has the mechanism to control. The only consequences are seen when the person is unable to control this, or when there is excess of or high rate of these processes, and that happens due to the excess of glucose or glucotoxicity. So glucose can be auto-oxidized, or glucose enters another pathway that is known as polyol pathway, leads to generation of sorbitol and fructose, and then methyl glyoxal or 3-deoxyglucosone, or what we have seen in the previous, that glucose binds to amino group of the proteins, leads to the formation of CIF base, that leads to the production of Amadori product, because this was the first person who discovered this ketamines and aldomines. So they are, and this Amadori products are further converted to. The below part shows that the time which takes for the formation of this. So the CIP or base formation takes place in hours to days, and Amadori days to weeks, and the formation of AGEs takes place in several weeks or several months. So this is very important in terms of like the proteins. Which are the proteins which get affected? So as we can see, the proteins which have longer half-lives or shelf life in the body are most prone to get glycated. For example, albumin, hemoglobin, and collagen, and other proteins. The proteins which have very short half-life, they do not get glycated because of these reasons. Even if they are glycated, the half-life of those proteins is very, so they are degraded by the body. So when the hemoglobin is glycated, we all know that hemoglobin is part of RBC. So when the hemoglobin, when there is high hyperglycemia, the glucose enters the red blood cell, or so what happens? Hemoglobin is glycated. So that leads to all of this, several consequences. So the glycation can be under several stages, like early, intermediate, and late. So early stage, the reaction starts with interaction between the carbonyl group of glucose or any other sugar and amino group of the amino acids of the protein. So as we all know that when we recall the structure of the amino acids, the two amino acids which have an additional amino group are lysine and arginine. They are involved in the glycation reaction, because other amino acids have the amino groups, but those amino groups are involved in the formation of peptide bond. So the amino acids which are involved in the glycation reaction are lysine and arginine, and the N-terminal amino group of the proteins. So this interaction leads to the formation of CIF base and formation of further rearrangement of this CIF base leads to early glycation products. So we can see here that sugar and proteins, they react, CIF bases form Amadori products, and so intermediate state is the formation of dicarbonyl. The examples are glyoxal or methyl glyoxal or deoxyglucosone, and the late stage is the formation of advanced. So intermediate stage is the interconversion of or formation of Amadori or Hens products. So examples include this. So the late stage of this glycation is the formation of advanced glycation end products. And in our day-to-day event, we can see this reaction happening. And that is how this reaction was discovered by a scientist, which I'll show you in the lecture. that when we cook the food, when we cook or when we bake or when we fry, the food items become brownish. That is because of the reaction between the, so the food, the browning of the food is a concern of food industry. So the glycation process occurs continuously in the body, even at the normoglycemic levels. Like, I mean, those who are non-diabetics, they also have this glycation. But the damage or consequences of this glycation or the harmful effect of glycation is realized only when the glucose concentration remains high for very long time. So that condition is hyperglycemia. So the amount of glycated albumin decreases when blood glucose levels are low, and the increase when blood glucose levels are. So you can see that the amount of glycated albumin is low, and the, so this is we all know that those who are diabetics, they go for routine sugar checking, what we call as fasting and postprandial. So that gives an idea that whether the person is hyperglycemic or not. But many doctors contest that, and they say that, okay, that doesn't give, because there may be other factors which lead to an increase in the blood glucose level. So what people do, they rely on estimation of another factor that is known as HB1AC, that is known as glycated hemoglobin, that gives an idea of whether that the glucose level is high for, has been high in that patient or the person for several months. For example, if the level of glycated hemoglobin is high or above 6 is the normal level of HB1AC. If the level of glucose HB1AC is more than 6, it means that individual person had a history of high glucose level for more than 3 months. Similarly, if the glycated albumin is high, it gives an indication that that person had high glucose level for several weeks. On the other hand, blood glucose level can be high blood glucose level can be an indicative of high sugar level for only last several hours. Okay. But again, every test has its own advantage and disadvantage. We cannot come to conclusion unless we do some other test. So the concentration of glycated albumin and hemoglobin can be affected because of the nephrosis, cirrhosis, and thyroid functional disorder. Similarly, glycated hemoglobin concentration can be altered or can be affected by anemia, renal failure, or cirrhosis. So this is what I was talking about, the how glycation was discovered. It was discovered by Louis Maillard in 1912 when he was synthesizing amino acids, when he heated amino acids and reducing sugars together. That resulted in a color change to yellowish brown. So the process of glycation is also known as Maillard reaction, and that is the term which people use in food and industry, that Maillard reaction means browning of food stuff. Okay, and that happens because during the cooking, so it affects the quality of the products also. So now I'll talk about some of the features of advanced glycation and products. So what are they? They are the end products of the glycation. They are very heterogeneous group of molecules. How are they generated? The three main routes for the formation of AGs are auto-oxidation of sugars, means the sugars which are there in the body, because they are reducing, so they undergo oxidation. Second one is Amadori rearrangements and the Schiff-based formation. These are endogenous sources of AGs or glycation products. There are exogenous sources of advanced glycation products, that is through the ingested food And that has become again a major consequence when we eat fast food and other things. We also consume lot of glycated products. So even a person is non-diabetic, the glycated products can enter our body and cause harm to. So intestinal absorption of exogenous agents is affected by digestive enzymes and microbial flora in the gut. So this is what we can see that it happens. So extracellular formation, Maillard reaction, glucose plus proteins. Okay, lipid peroxidation. Lipid molecules in the membrane are very prone to uh, attack by the free radicals. So lipid plus ROS oxidation, reactive carbonyl groups, lipid plus. So intracellular, extracellular, both that happens. Intracellularly, we have glycolysis pathway, as we have seen earlier, that glucose after this glyceraldehyde 3-phosphate step, it can get converted to 3-deoxyglucosone and so on. So whatever the route may be, or wherever it may generate, it may enter the circulation and bind to the proteins non-enzymatically and cause damage to the proteins and other biomolecules. So whatever the source may be, endogenous, exogenous, intracellular or extracellular, they have some common features and on the basis of that they are classified. They cause cross-linking of the proteins and some of them are fluorescent. So some of them are fluorescent, some of them are non-fluorescent. And so examples of this advanced glycation products, although we can see that the work started in 1912 by Maillard, then he worked on, and then many people worked on that. But unfortunately, this has not come to our textbook level. But those who study on this diabetes and work on glycation, they are aware, they know these terms very well, that carboxymethyl lysine, carboxyethyl lysine, pentositine, they are the biomarkers in the body for the ages. So if you can estimate this product CML or CEL in the body, one can get an idea about the accumulation of ages in. And so this is what again you can see here, that these are the structures. These are, some of them are fluorescent. So fluorimetry can be used to identify them. So what are the factors which affect age production? These include oxygen, reactive oxygen species, and redox active transition metals. Environmental factors like diet and smoking also influence the production of ages in the body. Now, what role ages play in the diabetes? So, as we know, high glucose can lead to the generation of or increase in the production of mitochondrial ROS that leads to oxidative stress, which leads to beta cell dysfunction. The other route of glucose acting through is the formation of ages, which binds to a receptor or which belongs to immunoglobulin family, and this is known as RAGE or receptors for age. Okay, so binding to or formation of age-RAGE complex leads to ROS production, that is reactive oxygen production, that leads to activation of NF-kappa B, and finally to inflammation and insulin resistance. So these are the probable mechanisms by which ages cause damage to the or ages amplify the condition under the diabetes and lead to the secondary complications of diseases. So this is what major organs or major parts of the body which get affected. Eye lens, we see the role of ages in the cataract or retinopathy. We have this blood circulation. We have collagen getting affected due to the accumulation of ages. we see in urine, we see in the cartilages, and so on. So, brief about this hemoglobin glycated, because uh, those who have read about diabetes, they might or sometimes we see the patient blood sugar report or blood report, we come across this HbA1c, that is known as glycated hemoglobin. So it accumulates in the body as a result of diabetes, or what we can use the term, that is hyperglycemia. So the concentration of HbA1c provides a glycemic history of the previous 120 days. So that has been a very good pathological test, which gives an idea about the hyperglycemic condition. So I mentioned in one of my slides that if the level of HbA1c is more than 6, it means that person had a high blood glucose levels for more than 3-4 months. So and if that person is undergoing any kind of treatment, and if the blood glucose level of the HbA1c level is less than 6, it means that whatever the medication is given to that person is working efficiently. There are still many controversies with this. I'll not go into that. So we can see that this is the normal. You have glucose entering into the RBCs. They interact with the glycated hemoglobin, and then then the function of hemoglobin, which is to carry oxygen, gets impaired. So that's another way by which. So what are the pathophysiology? Where is the glycation implicated? Aging, diabetes mellitus, Alzheimer's disease, Parkinson's disease, cataracts, retinopathy, arteriosclerosis, and increased susceptibility to infection because of the. So a cartoon, glycation causes heart disease, kidney disease, dementia. So these are all known as secondary complications of diabetes, because the high blood glucose level or hyperglycemic condition leads to alteration of the biomolecular function. So at molecular level, toxicity of glycation can be through the inhibition of functions of biomolecules, through precipitation of proteins, cross-linking of proteins, aggregation of the proteins, ROS and RNS generation, and strand breakage of DNA. So these
Excuse
are
me,
the
sir.
Yeah.
Excuse me, sir. I'm afraid we are running out of time, so you may please.
Yeah, I'm winding up.
if you
I'm
can
just winding up.
yes.
I'm coming to.
Thank
Yeah,
you, sir.
sir,
Thank
I'm coming to. Yeah. So these are the mechanisms by which glycation toxicity is suppressed. We have in our body, we have some enzymes, aldehyde reductase and glyoxylase. We have protein degradation systems. We have macrophage systems, and some drugs have been discovered, that is aminoguanidine. And people are working on anti-glycation agents and antioxidants for this purpose. So how do we protect ourselves from ages? Reduce consumption of sugar.
Sir,
Eat.
sir,
Yeah.
it is now running over, so you please conclude it within 1 or 2 minutes, sir.
Yeah. I'm just. So this is
Yeah, it is
just the
already
last one.
over.
Yeah.
Thank you.
Yeah. So we have to reduce the consumption to see the. So these are the ways by which ages are. So thank you, sir.
Thank you so much, sir, for